this week we will learn about the structure and the functions of nucleus and also other organelles inside plant and also animal cells. So first we're gonna look on the most important organelle, which is nucleus. So nucleus is where the DNA located, and also it is contain the cell genetic instructions. So terms that usually related to nucleus are nuclear envelope, nuclear membrane, nucleolus, nuclear pole, nuclear lamina, and also chromatin. So let's look on how does it look like. So the inner core of nucleus is known as the nucleolus. So we have the chromatin here, and it wrapped by the nuclear envelope that consists of inner membrane and also outer membrane. And also we have a nuclear pore here. So bear in mind that the nuclear envelope is connected with the endoplasmic reticulum, in which I will discuss about this one later. So nuclear envelope is the one that encloses the organelle, enclose the nucleus, therefore separating the content inside the nucleus from the cytoplasm, the liquid outside the nucleus. So nuclear membrane is a double membrane which is consists of inner and also outer membrane, and each membrane consists of a lipid bilayer. So this is how the surface of the nuclear envelope look like. As you can see here, nuclear pore is very obvious. Okay, and this is a nuclear lamina. Okay, the one that located on the surface of the nuclear envelope. So the nuclear pore function to regulate the entry and also the exit of molecules from the nucleus. And the nucleolus, which is the inner core of the nucleus, is located within the nucleus, and that is the location for the synthesis of ribosomal RNA. And the shape of the nucleolus is maintained by nuclear lamina, in which this nuclear lamina is composed of protein. So here. And how about the chromatin? Okay, this chromatin. So chromatin actually come from the DNA. As it contains DNA, as you can see here, the double helix of the DNA will wrap the protein histones. One is wrapped, the protein histone is known as the nucleosome, and the stake of the nucleosome here is known as the chromatin fiber, and the chromatin fiber will coiling with other coiling of chromatin fiber, forming the chromatin, and then the chromatin will coil again within the chromosome, and this is how the chromosome looks like. Okay, so in the nucleus, DNA is organized into discrete units that call as a chromosome. We have X chromosome and Y chromosome, as you can see over here. Okay, so each chromosome is composed of a single DNA molecule that associated with proteins. The protein that I mentioned earlier, which is histone, and the DNA and histone are together. When it's called together, it is called as a chromatin. So when cell prepares to divide, chromatin will condense to form a discrete chromosome, which is chromosome X and also chromosome Y. So now let's look on the structure and also the functions of other organelles. So remember last week we learned about cell fractionation, in which it is a method to separate major organelles from one another. So we won't talk about this one because we already learned it last week. Okay, so what is organelle? Organelle are the specialized structure within a living cells, either prokaryotic or eukaryotic cells, that carry out a specific, either in plant cell or animal cells, that carry out specific functions. So There are four major types or group of organelles, in which the first one is the ribosome, the second one is the endomembrane system, the one that consists of a membrane, the energy processing organelle, including the mitochondria and also the chloroplast, and last but not least, we do have cytoskeleton. So this is how the animal cells look like. Okay, we have the nucleus here, the endoplasmic reticulum, either rough or smooth endoplasmic reticulum with or without the ribosome, the Golgi apparatus, the lysosome, mitochondria, peroxisomes, microtubule, and so on. So this is how the plant cells look like. It consists almost all of the organelle that contain inside the animal cell, except for it do contain a chloroplast that is responsible for the photosynthesis process. So this table here show the presence of organelles in animals and also plant cell. Both of animal and plant cell consist of ribosome, endoplasmic reticulum, Golgi, vacuoles, nuclear envelopes, plasma membrane, mitochondria, peroxisome, and also cytoskeleton. Except for lysosome. Lysosome you can only find it in animal cell. Meanwhile, chloroplast only can be found in plant cell. Okay. So first, we're going to look on the ribosome. Ribosomes are particles that made of ribosomal RNA and also protein. And the function of ribosome is to carry out protein synthesis in two locations, either in the cytosol, which is referring to the free ribosome, or on the outside of endoplasmic reticulum, or nuclear envelope that known as the bound ribosome, because it's attached to other organelles. That's why it's known as a bound ribosome. So as you can see here, the free ribosome in the cytosol, and this one is endoplasmic reticulum, and we do have a ribosome that attached to the endoplasmic reticulum, which is known as bound ribosome. And this is how the ribosome looks like. It do have large subunit and also small subunit. So next, we're gonna look on the endomembrane system that consists of endoplasmic reticulum, Golgi, lysosome, vacuoles, nuclear envelope, and also the plasma membrane. However, we will learn about plasma membrane next week. Okay. So endoplasmic reticulum. So the endomembrane system. Actually, the main function is to regulate the protein traffic and also perform metabolic functions in the cell. So they are very complex and dynamic player in cell compartmental organization that I already explained last week. So the components of endomembrane systems again are including nuclear envelope of the nucleus, Golgi, lysosome, vacuoles, and also the plasma membrane. So all of these components are either continuous or they are connected via transport molecules, which is known as the vesicle. So first, endoplasmic reticulum, the biosynthetic factory. So endoplasmic reticulum account for more than half of the total membrane in many eukaryotic cells, and its membrane is continuous with the nuclear envelope that just like I mentioned earlier, and there are two distinct regions of ER. If the ER contain or bound with ribosome, it is known as rough endoplasmic reticulum. But however, if the surface of the endoplasmic reticulum is lack or no ribosome at all, it is known as a smooth endoplasmic reticulum. So here is a nuclear envelope, and nuclear envelope will extend it, okay, forming the endoplasmic reticulum, which is rough endoplasmic reticulum consisting of the bound organelle, and smooth endoplasmic reticulum is free or length from the ribosome. So what is the functions of smooth and rough endoplasmic reticulum? Smooth endoplasmic reticulum synthesize lipid, metabolize carbohydrate, detoxify drugs and also poison. Last but not least, to store the calcium ion. Meanwhile, rough endoplasmic reticulum has bound ribosome, which later secrete glycoprotein, in which glyco is referred to the carbohydrates and protein is referring to the protein. So the protein are covalently bonded to the carbohydrate. And then, since the rough endoplasmic reticulum contain a secret glycoprotein, they will distribute the glycoprotein through by using transport vesicle, and also it is a membrane factory for the cell. Next, Golgi apparatus, the shipping and receiving center. So Golgi apparatus consists of flattened membranous sac that called as a cisterna. As you can see here, cisternae, okay, the folding of the Golgi apparatus, in which the Golgi apparatus have two sides. The side that receiving the vesicle is known as the cis-phase, and the side that will shipping out or forming the new vesicle is known as the trans-phase. Okay, so the functions of Golgi apparatus are to modify the product of the endoplasmic reticulum, and then they will manufacture certain micromolecules such as the protein, and also sort and package material into transport protein through the transphase. Okay. Next is lysosome. Lysosome, the digestive compartment. So lysosome is a membranous sac of hydrolytic enzyme. They contain hydrolytic enzyme that can digest macromolecule. Therefore, their function is to hydrolyze the protein, the fat, polysaccharide, and also nucleic acid. Lysosomal enzyme work best in acidic environment inside the lysosome. Okay, so this one is lysosome. It is a membranous sac that contains lots of hydrolytic proteins that be enclosed by the lysosomal membrane. Okay, so inside the lysosome, they contain a lot of hydrolytic enzyme such as phosphate ester, nucleolase enzyme, protein digestive enzyme, carbohydrate digestive enzyme, and also lipid digesting enzyme. So some types of cell can engulf another cell by phagocytosis. So this will form a food vacuole. So once the cell engulf the food and forming a food vacuole, lysosome later will combine with the food vacuole, and because of lysosome contain hydrolytic enzyme, it will help the cell to digest the food inside the food molecules. So the lysosome use enzyme to recycle the cell on organelles and micromolecules, and that process is known as the autophagy or self-eating of cell on damaged organelles. So if there is any damaged organelles or unwanted molecules inside the cell, lysosome is the one that responsible to digest it or to remove the damaged organelles. So as you can see here, the phagocytosis, how does it look like? Food vacuole, okay, that contain the food through the process of phagocytosis will fuse with lysosome, and the digestive enzyme of the lysosome will help to digest the enzyme inside the food vacuole. And the other process is known as the autophagy, in which the lysosome, okay, will fuse with the organelles that being damaged, and they will digest the damaged organelles. So next, vacuole, the diverse maintenance compartment. Plant cells or fungal cells may have one or several vacuoles that derive from endoplasmic reticulum and also Golgi apparatus. There are three types of vacuoles. We have contractile vacuole, central, and also food vacuole. First, contractile vacuole found in many freshwater protists like amoeba and also paramecium. Their function is actually to pump the excess water out of the cell. So usually you can find it in the prokaryotic cells such as the protists. And then we do have the central vacuole. Central vacuole can be found in many mature plant cells, in which its function to hold the organic compound that being produced through the process of photosynthesis, and they will help to absorb water for growth of plant cells. Last but not least is food vacuoles. So food vacuoles can be found in many freshwater also, just like the contractile vacuole, in which it functions to contain the food from phagocytosis. Okay, here is the central vacuole inside the mature plant cell. And then as you can see here, here is the food vacuole, and then this is the contractile vacuole that used to remove the excess water inside the protist. So we already learned about the nucleus, rough endoplasmic, smooth ER, and also Golgi apparatus. Okay, next we will learn about the energy processing organelles involving including the mitochondria, chloroplast, and also peroxisome. So mitochondria is the place to change energy from one form to another form, in which mitochondria are the site of cellular respirations that including the glycolysis process, the Krebs cycle, and also oxidative phosphorylation. Meanwhile, we do have a chloroplast that found in plant and algae at the site for photosynthesis. Okay, and then peroxisomes are oxidative organelles. So let's look on the structure and functions of these three energy processing organelles. So mitochondria and chloroplast have similarity with bacteria. Why? Because these two organelles is enveloped or wrapped by a double membrane. They do contain free ribosome and circular DNA molecules, and they can grow and reproduce independently inside the cell. Okay, so mitochondria and chloroplast actually come from the endosymbiont theory, in which that our early ancestor, the early ancestor of eukaryotic cells, they engulf the non-photosynthetic prokaryotic cell, which form an endosymbiotic relationship with its host. So the host cell and on endosymbiont will merge into a single organism, in which it will become an eukaryotic cell with the non-photosynthetic prokaryotic cell, which is now known as mitochondria. Okay, so somehow some of the eukaryotic cells that already contain the mitochondria will engulf other photosynthetic prokaryote. Then, therefore, this photosynthetic prokaryote later will live inside the host, and now it's known as the chloroplast. So the next eukaryotic cell do contain chloroplast and also mitochondria. So let's look here. This is the eukaryotic cell. It engulfs the non-photosynthetic proteins, now becoming a mitochondria, and some of them uh, will engulf photosynthetic prokaryotes or proteins, and now it become a chloroplast. Okay, so therefore it become photosynthetic eukaryote. I will give you the link of the video of how does the endosymbiont theory occur later. Okay, here's the same. So mitochondria is a chemical energy conversion place for chemical energy conversion. So almost all eukaryotic cells have mitochondria. They have smooth outer membrane and inner membrane that folded into cristae. Cristae present a large surface area for enzyme that synthesize ATP, meaning to say they do contain ATP synthase protein to synthesize ATP. And the inner membranes will create two compartments, either intermembrane space, the space between outer and inner membrane, and the mitochondrial matrix, okay, the fluid that being enclosed by the inner membrane. Okay, so some metabolic steps of cellular respirations are catalyzed in the mitochondrial matrix. Let's look here. We have outer membrane, and the space between the outer membrane and inner membrane is known as intermembrane space, and the fluid inside the inner membrane space, or the one that be enclosed by the inner membrane, is known as the matrix. Okay, and the folding of the inner membrane is known as the cristae. So next, chloroplast, the site to capture light energy. So chloroplast can be found in leaves and also other green plants, and plastid are known as plant organelle. For example, a plastid, which is actually it's referred to a photosynthetic prokaryote with green pigment, is called as a chloroplast. So chloroplast is plastids. So the function of chloroplast, of course, it contains green pigment, which is known as chlorophyll, and it contains other enzymes that function in photosynthesis to make food for the plants. And their structure includes thylakoid and also the stroma. So as you can see here, it do have a double membrane too. We have the outer membrane, inner membrane. The space between outer and inner membrane is intermembrane space, and inside here is known as a stroma. Okay, and then one of this thing is known as a thylakoid, and one unit is known as thylakoid. A stack of thylakoid is known as the granum, and this junction is known as a lamella, and inside the thylakoid is known as a thylakoid lumen. So next, peroxisome, a place for oxidation take place. So peroxisome are specialized metabolic compartment bounded by a single membrane. They produce hydrogen peroxide and help to convert it to water, and it performs reactions with many functions. However, The connection between peroxisomes and other organelles is still unknown, and the functions of the peroxisomes are to neutralize the harmful toxin, to carry out the lipid metabolisms, and also to carry out, of course, the oxidation reaction that will break down the fatty acid and also the amino acid. So this is how the peroxisome looks like. They do have a plasma membrane that made up from a lipid bilayer and crystalline core. Last but not least for today, cytoskeleton. There are three types of cytoskeleton. We have microtubule, microfilaments, and intermediate filament. Not myofibrillia. Myofibrillia is not cytoskeleton. So, cytoskeleton is a network of fibers that organize structure and also the activity inside the cell. It extending throughout the cytoplasm, therefore its content only at the cytoplasm, not inside the nucleus or nucleoplasm. So the function is to support and motility. So inside the cell itself, vesicle can travel along through the monorails that provided by the cytoskeleton, or it looks like a road that to guide the vesicle to find its way. And also it will help to regulate biochemical activities, to support the cell and maintain its shape, and also it will interact with the motor proteins to produce motility. So for example, we have the microtubule, okay, that will work together with the motor protein function for the motility inside the cell. So as I mentioned earlier, there are three types of cytoskeleton. Fiber, we have microtubule, the thickest of the cytoskeleton, the thickest component of the microskeleton. Then we have microfilaments, or which also known as actin filament, which is you can find also it's very important for muscle contraction. And these microfilaments are the thinnest components of the cytoskeleton. And last but not least, we have intermediate filament, fiber with diameters in a middle range. So the thinnest one is microfilaments, then intermediate filaments, and the thickest one is microtubules. So let's look on the structure of the microtubule, the thickest one. So it contains tubulin dimer that consisting of alpha and also a beta dimer. Its diameter is about 25 nanometer, and it contain they have a hollow tube here, and it main function to maintain the cell shape, motility, chromosome movement, and also organelle movement. Next is microfilament. As you can see here, microfilament diameter is about 7 nanometer, and it made up from the actin protein, actin subunit, and function also to maintain the cell shape, changes in cell shape, muscle contraction, cytoplasmic streaming, cell motility, and cell division. And last but not least, we have intermediate filament, the fibers. This is the fibers. Protein supercoiled into a thicker cables. It do not have a hollow, and its diameter is about 8 to 12 nanometer. And one of the protein that make up the intermediate filaments is referred to the keratin protein, which also can be found inside your hair. And its main function, of course, to maintain the cell shape as the anchor for the nucleus and also other organelles, and It is also responsible for the formations of nuclear lamina on the nucleus. So in many cells, microtubules, let's look on the microtubule. Microtubules, which is they're made up from the tubulin polymer, the thickest cytoskeleton, grow out from a centrosome near a nucleus. You can find this during the prophase stage of mitosis and meiosis, and that's why centrosome is known as a microtubule organizing center. So in animal cell, the centrosome has a pair of centrioles, each with 9 triplet microtubule arranged in a ring. So how does it look like? It does look like this. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplet, this is 3, right? 9 triplet sets of microtubules and 2 sets of inside a new in a new centrioles. So these 2 centrioles will make a centrosome that is responsible during the process of meiosis and also mitosis. Remember? Each centriole contains 9 triplet sets of microtubule, and these 2 centrioles will known as a centrosome. So next, cilia and flagella. Microtubules, as I mentioned, it's for the cell motility, right? So it does control the movement of cilia and also the flagella. So cilia and flagella are very different in their movement patterns. However, they do share a common structure in which they have a core of microtubule that's sheeted by a plasma membrane, a basal body that anchor the cilia and also flagella, and they also have a motor protein called as a dynein, in which dynein drives the bending movement of the cilia and also flagella. Okay, as you can see here, this is how the motions of the flagella looks like. You can find it, one of the example is you can find it in the sperm cell. And then we also have a cilia, in which the movement is backward and forward. We have a power stroke movement and recovery stroke. Cilia, you can find it in your esophagus and also in the fallopian tubes of female reproductive organs and also the other parts of our organs. Okay, this is how the cross sections of the cilia look like and also the flagella. As I mentioned earlier, they contain a basal body. Both cilia and flagella have a basal body, and also both of cilia and flagella are wrapped by a plasma membrane. And the third one is both of cilia and flagella do contain dynein proteins, okay, which is responsible for the movement patterns of the cilia and also the flagella. So how does the dynein protein helps to control the movement pattern of flagella and cilia? Okay, so dynein will grab, move, and release the outer microtubule, and then it will make the protein to closely limits its sliding, and the forces exerted by the dynein arms will cause doublet to curve, bending of the cilia and also the flagella. Okay, as you can see here, this is the dynein protein that connecting between the doublet microtubules. So when there is a ATP, so it will control how the movement of the cilia and the flagella. Okay, so either like this movement or in the flagella, or this recovery movement or the wave flag movement in the cilia. Okay, so let's look deeper on the functions of each of the cytoskeleton. Next is the microfilament. Microfilament is the thinnest components of the cytoskeleton, in which it functions for the cell motility. And in our muscle cell, thousands of actin filaments are arranged parallel in one another because they are the very important components for muscle contraction. So as you can see here, this is the actin filament, okay, and they do also contain a thick filament here, okay, that have a myosin head that will attach to the actin on the actin filament, thus producing a movement. So, and also it is very important for the amoeba movement, in which inside the pseudopodia of amoeba, and it will extend and contract through the reversible assembly and contractions of actin subunit into microfilaments. And also it is very important for cytoplasmic streaming, in which cytoplasmic streaming is referred to the circular flow of cytoplasm within the cell, and this streaming speeds the distributions of material within the cell. And in plant cell, actin from the actin filament and myosin from the thick filament interaction and salt gel transformation drive the cytoplasmic streaming. See this one, how does it look like, the amoeba movement, and also cytoplasmic streaming movement, in which microfilament are the one that responsible for these two types of movement inside the cell. So last but not least, intermediate filament. Intermediate filament range in the diameter about 8 to 12 nanometer, as I mentioned earlier, and most of the intermediate filament composed of keratin fiber, in which it will help to support the cell shape and also the fixed organelles in place. And intermediate filaments are more permanent cytoskeleton fixture than the other two classes, microtubule or microfilament. So I think that's all for today, class. Remember, today we learned about the nucleus and also the organelles, including ribosome, and the organelle in the endomembrane system, cytoskeleton, and also energy processing organelles that involving the chloroplast, mitochondria, and also the peroxisome. So I think that's all for today, class. Thank you.